And today we're really excited to kick off our new Research Spotlight series, BME Faculty in Focus, where we're going to highlight research conducted by our Columbia BME faculty. Moving forward, these lectures will be available on our BME YouTube channel at youtube.com/ColumbiaBME. Throughout the presentation, if you have questions for our speaker, please feel free to type them into the Q&A area at the bottom of your screen, and we'll try to answer as many as possible at the end of the lecture. And at this time, I'm going to tell you a little bit about our inaugural BME Faculty in Focus speaker, Professor Ed Guo. So Dr. Guo is currently Chair and Stanley Dicker Professor of Biomedical Engineering and Professor of Medical Sciences in Medicine. Dr. Guo directs the Bone Bioengineering Laboratory in the Department of Biomedical Engineering at Columbia, and his research focuses on micromechanics of bone tissue, computational biomechanics, and mechanobiology of bone. Dr. Guo's past honors include the Young Investigator Recognition Award from the Orthopedic Research Society, the National Research Service Award from NIH, a Career Award from the U.S. National Foundation of Science, funds for talented professionals from the National Natural Science Foundation of China, and the Christopher R. Jacobs Award from the Biomedical Engineering Society. Professor Guo was one of the founding members of the BME department at Columbia and has served as its undergraduate chair, ABET chair, vice chair, and interim chair, and he was elected department chair in 2017. During his current tenure as chair, the department has risen to the top 10 in the country. So welcome, Ed.
Thanks so much, Lisa, and thanks so much spending the Friday afternoon with you and starting off the 5 focus. I'm very happy to present my love, which is my bone bioengineering research. So I'm going to be talking about really the microstructure mechanics and also the mechanobiology. And at the end, you can see the structure, mechanics, and mechanobiology, everything actually is linked. And hopefully we can also move in beyond just what I do. So, you probably don't think too much about it. Why do we need a skeleton? Skeleton actually is quite ancient, right? So when you have shark, actually have bones, and then all the species come from the land, come from sea, and so therefore I think the evolution when we have crawling animals, have the amphibious animals, and they have mammals, the evolution process now I study more bones now, really understand is we're still in it's a human history, it's very short, so therefore we're still in a very long journey to overcoming the baggages we have because we're too heavy. So therefore we want shedding off as much as bone as possible, because when we first come to the land, you have two choices because you are at risk to eat by other species. So one strategy prevailed for quite a while, but at the end, by these unplanned events, they disappeared, right? So that's dinosaur. So the one way to do is to grow bigger and bigger, and therefore you cannot be eaten by anybody else. And that one failed because asteroid hit the Earth, and the mammals, humans, actually survived. But we took a different strategy such that we actually try to get rid of as much as bone weight as possible. It's very interesting. In the old bone biology evolved from fish biology to mammal biology, so you create new proteins. All the proteins is either tells you do not making bones or go to remove bones. So that's very interesting. Of course, we needed to have bones because the bone really supporting our every motions we have. And then this is my former student from the lab. She was a dancer before. So we are interested in the bone, and then particularly in our lab, we're interested in the bone microstructure and also osteoporosis, right? Showing here is actually a very nice picture of a skeleton in here. That's in the spine, vertebral bones. And then even by eye, you can see there are dedicated structures in there, right? On the right side here, you can see piece of kind of sheet of bones that look like a plate-like structure. And then in the middle, you can see, they have some things like single column or like a rod, that structure. So that's the feature I want to observe is they actually have kind of plate rod-like structure. Second one is even looks disorganized, but they do showing the axial direction and then all the bones along this way. So in osteoporosis, or when getting old, then you lost in bones. So that's most important, right? It's very apparent you lost some amount of bones. You look at this aging bone, it's actually not bad, but you can see how much bone they lost. But if you're observing also carefully, those clay-like structure all disappeared, replaced by mostly this rod-like structure. So the consequence, for example, is a fractures. And one type of fracture is called spine fracture. Actually, it's silent. You don't know about it. So if you look at this picture of a Chinese woman from Hong Kong, these are 3 generations of women, and you can see the grandma actually is bend her back, and then that's actually the consequence of spine fractures. You look at the X-ray on top, you see the 2 top and bottom vertebrae actually is a very nice square shape, and in the middle you can see the one actually skin reduced dimension actually is quashed. So that's a spine fracture, so causing the curvature, but most importantly also can compress the spinal nerve and causing disease, and also causing other cardiovascular features. Another thing we don't think about too much about bone, and it's still very challenging, is other disease is osteoarthritis. We still don't know what the cause of osteoarthritis, and then the treatment is essentially unknown. More recently, we begin to understand a little bit more biology now. So on the surface, that looks like opposite what we see in osteoporosis, right? So I'll show you in the bottom, a picture in here in the middle, assuming it's normal trabecular bone. On the left side is loss in bone in osteoporosis. And then in the end stage of osteoarthritis, or we call it OA, and you can see they actually have a lot of bone. So therefore it's like almost like a lot of bones. But now from recent study, we have found out the underlying cause of osteoarthritis may come from actually abnormal resorptions in here. So therefore, that's the understanding here. But showing right in here a picture, we call it end stage OA because you can see the medium side of this knee joints here is red because they don't have the shining cartilage anymore. But notice on the lateral side, on the left, actually you can see still maintains the good cartilage you have here. But it's so painful that the patient is ready to do a total joint replacement. So another thing really fascinating for our lab and for many people working bone is live. They're actually able to adapt into how much you use a mechanical. So we have Australia Open going on right now. This is a picture of US Open. You can see the playing arm is not only muscle-wise stronger, the bone actually is 20 to 30% bigger than the non-playing arm. So there is a hypothesis which is very old, ancient, and is more than 100 years old. It's called Wolff's law. Essentially, it's bone mass and microstructure follows the mechanical laws. But what the underlying message is not known. And then we reactivate space activities now, and of course in the mask is really put a lot of news into flying to space, and in space you lost in bones, right? And then you lost in about 2% of bones per month if you stay in space. So therefore eventually we go to Mars, and then we facing tremendous problem of 1 year flight to Mars. If you're not able creating this super centrifuge, if you will, and they're going to lose a lot of bones. And then you can see loss in 24% bones sounds like not too bad, but the consequence in mechanical property bone is actually very devastating. So we are interested in bone microstructure. Fortunately, there are technology available to study this microstructure non-invasively. It's called the microcomputer tomography. It's CT machine, but it's just high resolution. And they can also use this one in patients. We just give them a different name. It's called high resolution peripheral quantitative computer tomography, HR-PQCT. You can see right side is a scan of the tibia. You can see, kind of representing the microstructure in there. So as I mentioned, I'm very fascinated by microstructure, right? So I'm particularly interested, long time ago, is can we separating these nice trabecular plates and rod-like structure, and they also have a way to quantify orientation. So with the research many years ago now, is we developed this technique. is called individual trabecular segmentation, called ITS. You can see if you zoom in that three-dimensional image you see a single rod like trabecula, and then it's voxel you see here. And by this segmentation technique, you can see this yellow single rod like trabecula has been segmented automatically from structure. So this segmentation is automatic, do not really need human intervention. And therefore now you can describe the microstructure differently. So for example, we separate them into the green showing the picture in the right, that's plate, and the red is showing picture on the right is rod. So therefore you can count how many plate are in the bone. It's called plate bone volume fraction. So bone volume fraction is essentially bone volume divided by total volume. So if it's plate, we put a P in front of them. If it's rod, we'll put an R in front of them. So that's a new description of how much plates in there, how much rod in there. And then you can quantify now, have technique, quantify individual, each one of them, what's orientation. So therefore, you can define them as a longitudinal along the anatomic position, anatomic directions, or transverse, in perpendicular to the anatomic directions, right? And then you can also, so if we measure the orientation, what we found out, even though it looks very disorganized, but all the bones, microstructure bones, trabecular bone in your body actually are designed this way. So you can see the plate mostly in the longitudinal direction, and the rod mostly is in the transverse direction. So therefore, the trabecular bone is designed such that the plate are aligned with where the anatomic location directions or loading directions, and the rod is almost like a rebar in your building to reinforce them, right? And then you can also looking at the distribution of the thickness, and then you can also count number of plates numbers in here. So I want to look at there's something here now. It's the total amount of plate or rod depends on two factors, right? One is individual thickness, another one is number. So therefore, roughly, the number times the square of thickness or so on and so forth is equal to total bone volume. So therefore, you can see the two ways you describe the bone volume fraction. One is number, another one is thickness. So this may come into play later on, important. So the most important thing about the trabecular bone is the mechanical property highly depends on the, is that the animation? No. Okay, that's very simple. So the mechanical property plotting in here is a Young's modulus in the longitudinal direction or in the axial direction, and plotted on the right, the horizontal axis are three parameters: is bone volume fraction, plate bone volume fraction, and axial bone volume fraction. Axial bone volume A means the bone volume fraction in that direction, right? Because we're talking about E3 is in the vertical direction, for example. So you can see one thing is how much bone you have in there describe mechanical properties. They can range in from just 100 megapascals to 1,000, 3,000 megapascals. All depends on how much bone volume you have. And then they describe most of them, right? So R squared is 0.94. Out of those bone volume fraction, you can see if we only measure the plate bone volume fraction, which PBVTV in green, it actually equally describes the mechanical properties. And if we only counted the bone volume fraction in the direction of the mechanical property, they almost completely describe the mechanical property. The R squared is 0.99, so therefore there's not really too much variation, cannot be explained by bone aligned in this direction. So therefore, the mechanical property is really determined by how much those microstructure bones aligned in that direction. And then, of course, this is if you look at them, they look like plates, looks like rods, but they're not exactly plates or rods. That's biology is, right? So how we actually show is actually just spongy plates and rods. So we actually can using a experimental computation way to prove it, right? So you can see on top are original micro CT images. And then we can do this called ITS, and then creating a alternative model, the plates and rods. We call them PR model, right? So you can see you have the, you show in the middle, those are plate representation. And then biomechanically, we can also making an orange model, we call the voxel model. So therefore, there's no assumption of plate and rod. So they are original structures in here. And then we can do experiments of sample. You can see that sample was have done the experiments, and then that's in green. And then we model them. And the yellow was a voxel model, means do not make assumption of plate and rod. Another one is alternative model based on really the plate and rod segmentation. Note, in these two models, called FE, finite models, and then we're using exact same parameters. We're not cheating anything, right? So voxel model and prior model have the exact same thing. So therefore, you can see, in the linear version, this straight line here, they both voxel model and the PR model doing exactly the same, right? So therefore, and then if you look at when they begin to bend, that's where they begin to fail, and then you play the rod model, actually model a little bit better. So therefore, we can confidently say by doing this experiment, the trabecular bone biomechanically is described by plate and rod structures. And getting very exciting is, as I mentioned before, you have this HR-PQCT, they probably have about 50 of them around the world right now. Colombia, fortunately, have one. Is the original voxel size 82 microns, and the second generation actually 61 microns. But we actually can scan them at 43 micron as well, but in general, we don't do that. So you can see these are non-invasive. You can do on patients, and so therefore you can scan, the patient's radius and ankle, and you can see that's a picture of me in there. I think the bone, I don't know whether this one bone is me or not, but they can see you really can have a nice structure of this one here. So showing the application of this imaging technique as well as our ITS, showing here is a example of a scan. So you can see scout view showing the tibia and also radius, okay? And then you can see the normal patients, they really have nice structures. And then in some patients that have structure, you can notice them, they have changes in the trabecular bone microstructure in the middle here. Definitely you lost in bones, and then maybe hopefully there are also some other changes here. So therefore you can also creating a patient-specific model based on those images, and you can perform a virtual mechanical compressions, and they can determine the whole bone, what's the stiffness, or you can taking out only the trabecular compartment, look at the spongy bone, and also can calculate in the stiffness well. Right. So we have done many studies with my collaborator, wonderful collaborator from the Columbia Medical School and the Criminology department. And one of the example here is we recruiting patients who has postmenopausal women with vertebral fractures and without vertebral fractures, right? We age match them. As you can see, why 85% of them are Caucasian. The reason for that, 80% are African American, and then they have similar bone mass index. And then normally when you diagnose who has osteoporosis, have risk of fracture, you're doing a called DEXA, okay, measure the bone mineral density, and then its area is two-dimensional, right? So you can see, we know those people has spine fracture, but you can see the fracture one versus control, the lumbar BMD in here, LS, they actually are not different. Also, majority of score actually are above 0.25, because 0.25 are classified as osteoporosis, the T-score. So those people do not necessarily have osteoporosis, they have osteopenia, but they still do not they have fractures, and also the bone density measurement cannot differentiate whether they have fracture or not, right? So if you look at the HR-PQCT, that's the control sample at the left, and the vertebral fracture patient at the right. And then you can, as I mentioned, you can perform in the ITS, taking trabecular bone compartment, and they also can analyze the cortical as well. So for the trabecular bone analysis, we're showing here plate is green, rod in red. So by just looking at them, certainly in the fracture side, you have more rod, right? You have more red in here. And quantify them indeed, right? There are significant reduction in the fracture group with plate and rod bone volume fraction (PVDV), and then in both radius and the tibia. And if you calculate it, we call it PR ratio, plate versus rod ratio, then they can significantly have a reduction in the plate rod ratio, especially in the tibia. So therefore, that means there is a transition loss in plates, and then they actually do transition from plates to rod. Right, and then you also lost in the area of bone volume fraction. That's actually very interesting, because as I mentioned before, the axial bone aligned in that direction really determines the mechanical properties here. And then if you look at what the mechanism loss turned out, you lost him by significant number of plates. So P, TB, N, that's number of plates. And then you also lost him the connections, that's called PP junctions, PR junctions, RR junction. So the loss is by trabecular number and also connectivities. And then the consequence in here is you loss in the trabecular stiffness, how strong the trabecular bone is, and the whole bone stiffness. So therefore, with computation as well as the technology, we are able to identify what the the significant difference, much more sensitive than the density measurement. So in summary, in the osteoporosis and fracture, we are going to summarize. In general, it's loss of trabecular plate, loss of axial trabeculi, and then you have plate rod conversion, right? And they also lost in a trabecular connectivity. So let me go to the other issues we're working on the lab. That's actually most in the past 80 years is working on osteoarthritis. As I mentioned, there was a recent biological study showing, for example, in ACL tear, those patients will have early osteoarthritis, and then the first thing happens is bone resorption. So how the bone resorption initially leads into eventually a lot of bones in there is not understandable, right? And then so we begin to apply our technique in the subchondral bone, means the bone underneath the cartilage, right? So showing here on right is a result for femoral neck, which we talked about before, right? And you can see this is a distribution of rod versus a plate. You can see the rod on the top here is most of them is transverse directions. And in the bottom here, the plate mostly is the longitudinal direction. And then also want you pay attention to the numbers we're showing here, right? These are thousands, 25,000. These are 6,000. So therefore, in the femoral neck, you have many, many more plates than the rods, right? It's plate dominant. So looking at the left side is really the subchondral bone underneath the knee, the tibia, right in here. And then the rod distribution is very similar. They're actually, most of them are transverse directions. And if in place, and they can see there was a predominantly in the longitudinal direction, then also there's one close to transverse direction, also have another kind of, peak around there. So therefore, it looks like there is like the hammer has two humps. And also I want to point you to the numbers in here. In here, the rod is about 1,000 and plate is 400. So therefore, subchondral bone is different from the femoral neck or from spine because they are more rod and then they have less plate. And then the plate distribution maybe have some unique features in here. So as we mentioned before, the early study we have done is taking some images from China. And now we actually have done our own studies, we just need to writing them up as well. This is doing the total knee replacement. You can take in the tibia plateau cutting off in here, right? Showing here in the medium side, you can see almost no cartilage. And then on the lateral side, actually it's mild. And then you can see they actually have some shining cartilage in there, right? And then we can scan them, and then we're going to study those subregions in here. Right, and this is a cross-section picture of the micro CT images you have after section. So you can see on top is control samples, normal, we call them normal, okay? And then the bottom one is always sample here. Once again, you can see on the medium side, there was a lot of bones in here, right? And then even though this is X-ray, CT, but you can also see the shadows of cartilage, right? On top of here, you can see there's a cartilage really distributed nicely in here. And so therefore in here, definitely in the medium side, they almost lost complete cartilage. But interesting, on the right side, which is lateral side, and then they are not normal, okay? Absolutely not normal, because you see the right side here is a thing going up. That's called osteophytes. And they can see the subchondral plate here is not normal because we call subchondral plate fracture because they lost something, they have fracture here, right? And then, but they actually have 4 thickness. You can see here, the cartilage actually 4 thickness here, even though they're not all normal. If you look at the bone, subchondral bone here, apparently there's not too much difference into here, right? And then, indeed, if we take in those lateral side, and then you do histology, classify their cartilage integrity, you can see those regions, external, anterior, posterior, they actually looks normal. okay, compared to the histology here. So if you do a scoring here, those areas actually are not necessarily have OA, right? So therefore, that's not significantly different from the control samples in here. So we're going to focus on this area, and then to look at the measurements we have. So you can see these are traditional measurements we have. You know, if you look at bone volume fraction, in those one, they have a intact cartilage thickness, and the bone volume fraction is no different. And then in here, I'm also reporting the plate bone volume fraction is different. So that's where the troubles are having here in the past, right? And then I shaded area here is actually those places they do classify as OA. But in the meantime, they have differences in bone volume fraction. They also have differences in plate bone volume. I don't want to, you focus on that. Want to focus on in OA sample, but the cartilage is intact, right? So therefore, when you have OA, you look at bone and cartilage. They either have, in the previous technology is showing, you either have bone density change, you have OA, or you do not have a bone density change, you don't have OA, right? So therefore, that's why it is difficult to figure out which one is first. But because we now separate the plate and rod, right? So therefore, you can see now, and then if you look at the mechanical property, sorry, if you look at the mechanical property, they're also in these regions normal, they do not have difference. But now because we separate them in the plate and rod, so therefore you can see now, they actually, even though they looks normal, bone volume fraction is normal, but they actually have significant low rod bone volume fraction in the OA group. And then the PR ratio has been changed. So the plated rod ratio has increased significantly in the OA samples, right? Even though you cannot measure by the traditional bone measurements here. And then the loss of rod bone volume fraction is by the bottom here is number of rod. Rod number has been significantly decreased. It looks normal, but because of this ITS, we've already shown that, right? And then the reason not been show there was differences in bone volume fraction because when you lost in the rod, the remaining rods actually become thicker, right? You can see that actually a significant increase of rod numbers. That's why if you just measure how much bone volume fraction they're having here, they're not able to pick up by that. Now, if you look at the medium side, everywhere is osteoarthritis. The score is all different. And of course, now the bone volume fraction shows up now is very, very different. And then the plate bone volume fraction is very, very much different. The other plate bone volume fraction here. And then the mechanical properties over the roof here. And now, interesting, most rod bone volume fraction still lower in OA, very consistently with the normal we're having here. Of course, the ratio between the plate and rod ratio is going off the roof. It can be 3 times, 2 times, quite a different. And then the deficit in the rod number is still remain the same, right? You can see they're still in the OA sample. They have less of a number of density. And the thickness, the remaining trabecular plates and remaining rod, they become thicker. Because you lost in the rod, the remaining rods have to grow thicker because they still have some mechanical forces in here, right? So that's our cross-section study that, this end stage ones here. So therefore, we have the new hypothesis. Maybe the loss of rod bone volume fraction actually occurs earlier in OA, right? So this one is supported by a guinea pig models, and then those nice looking guinea pigs you have, and then they actually unfortunately develop OAs, osteoarthritis, in 4 months and 5 months. So in here we look at 3 months. You can 3 months the cartilage is normal, so second month to 3 months they're normal, right? But if you look at using the ITS analysis, you can see in the second month and the third month, we already identified the actually lost rod in here. So therefore, we have a new hypothesis right now is the OA could come from initial loss. You can see, in subchondral bone, you have a very nice distribution of rod, most of the rods, some are plates, and for somehow you lost in the rod. When you lost in the rod, and then when you lost them, they're gone. You cannot really make them happen again. And then to remain, because of mechanical loading, so the plate becomes thicker and thicker. Eventually, they're so thick, showing here schematically, and then the causing distribution changes in the cartilage, and then leading into loss of cartilage, and then the loss of cartilage increases the bones. So you have the vicious cycle going on. So that's a new hypothesis we have right now. And then in order to test them, you need to really do a study. So the new version of HR-PQCT, if you're not too tall, if you're not too heavy like me, and then you actually can stuck your legs in there, you can actually scan your knee, right? So that is one is my knee, right? You can see, you can scan the femur, you can see the tibia, and then you can look at the really nicely high resolution here, right? Therefore, you can identify your regions you want to study in the tibia as well as in the radius. So we begin some studies looking at one of the diseases, and one of the injury have is ACL tear. So in ACL tear, more than 50% of these people will develop a early osteoarthritis in 10 years, right? And then we know before it's a loss in bones. You can see we have those people immediately after ACL injury, and then, you know, roughly 1 month later before the surgery, and another one is 1 month after the surgery replacement, okay? You can see during that period of time, you lost in amount of bone marrow infections, and the plate bone marrow infection remain the same. And then indeed, as observing here, using this technique, we identify that actually a loss in the rods initially, and also the plate rod ratio has been changed, right? So therefore, we have some evidence suggesting those early changes, even though it's very subtle, but they're actually causing fundamental structure changes in here, right? So I want to summarize this essence of trabecular bone is trabecular plates is just bunch of plates and rod, trabecular bone. And then the plate and axial trabecular are very important. In general, the supporting mechanical load. And then in OA, if too much plates, too much is strong in axial one, they cause a non-uniform distribution in trabecular bone. So this ITS morphology is really providing a morphological and biomedical mechanisms for us under the cell structure. And then in osteoporosis, you have plate loss, plate to rod conversion, and reduced connectivity. In osteoarthritis, you lost in rod, thickening plates, but also have reduced the connectivity. So too little, too much in bone microstructure are not good. So I'm going to talking about, and then I have the people asking me questions, you know, why you have plates? Where do the plates come from? And I was never able to answer them several years ago. So one of the things I mentioned before is that bone is alive to respond to mechanical loading, right? So therefore, you know, how to quantify the mechanical loading and able to quantify stress strain is challenging biomechanics problem. And also in the cellular level, how to respond, what's the mechanism is fascinating, right? So therefore in our laboratory, we have done animal studies, we have done explant studies with those single cells that really center around to try to answer these questions. And then before we get into some measurements, we're going to introducing how bone is formed, right? The bone dynamics. So the two processes going on in bone. One is called remodeling. Is you resorbing bone, is formation following resorption on same surface. This happens every day because that's where regular bone maintenance to usually balanced, and then make sure you actually always have new bone, so the old bone will be replaced by new bone, right? And then another process we call the modeling, but it's not a model, it's a modeling process, right? Those are either be uncoupled formation, just formation, there's no result before, and uncoupled resorption. We call them anabolic and catabolic, because catabolic lost in one here. In before, people in general believe that's during the growth and development. You can see right now we actually deserve it. You know, it's maybe not that true. And you can see both mechanisms can lead into bone loss by modeling and by remodeling. And both mechanisms can also lead into bone gain as well. You can see if you have catabolic modeling, you have a lot of bone loss. If you're remodeling, you're making bone less than what you resorb, you have bone loss as well. So, but that's a very important thing right here. So we want to determine these questions. Where and how much bone is formed and resorbed in response to mechanical loading, especially interested in whether it's on plates and rods, right? And then the second one is what mechanism, whether it's a modeling and remodeling, because I'm not going to go to detail, but what it means is this actually relate to the bone biology behind it. This modeling process and remodeling process are just different bone biology here. So we're using these mice tibia loading animal models, and then we applying non-invasive mechanical loading to the right leg and the left leg as long load control, and we do the loading for 5 days a week, and then we actually do many more weeks here. And then in order to quantify the, you know, we actually can use in the micro CT as a way to quantify the bone form. So therefore, we actually do a lot of the scans. We do weekly scans. You know, this week 1 is where started mechanical loading, and then loading them for 5 weeks, and then we, you know, and then do another scan in 6 weeks. And so you can see we're starting -1, 0, and all the way to 6 weeks. So we have a weekly scan, and then we'll measure one in trabecular bone, shown here, also can quantify the one in cortical bone, right? And then what we do is we will have so many different weeks, and then we do a registration. We put them all in a fixed coordinate system and register them. And then by compare each week by the images, you can actually quantify in red a bone disappeared, resolved. And also bone gained in green is formed, right? So bone formation and resorption between all the weeks in the same coordinate system. And then because you can track in every week, so now by tracking the bone voxel for 3 weeks, and then you can actually define what the kind of bone is formed, right? So if you see there was a background, suddenly you have bone formation, that's anabolic modeling because you're adding bone from nowhere, right? And then if I have resorption, become background, in 3rd week is still background, they're gone. So the carbonic modeling. And another one is if we see resorption in 1 week, this background means stop, and then in 3rd week that you have bone formation, that's a remodeling. Okay, actually I need it. Actually it should be just remodeling, not remodeling. All right. And then this one showing the example here, and then in this location you can see there's nothing here, suddenly have greens in there. So that's anabolic modeling. And then in here, you see one of the trabecula, the resorbed, and then it's actually completely gone. It's catabolic modeling. And then here, you have surface, is resorbed, becomes smaller, but then actually come back with the following weeks of formation. So therefore, you can separate the bone formation and resorption here. So this is just looking at a representative pictures you have here. Left side is unloaded. Right side is loaded. So definitely you have more patches going on in color, right? In the blue and in the yellow. That means a new bone formation. You can see that you have much more yellows in the loaded one and some in the blue. That's in the rod, right? If we quantify them, indeed, in the anabolic modeling, 1st week, 2nd week, 3 week, and 4 week, and you can see the significant plate. Significant bone formation by anabolic modeling on plates, but not on the rod, right? So that's what it is. When you begin loading them, they're actually adding anabolic new bones in the plate, not on the rod. And then you can separate them into different orientations, and very interesting, in axial, oblique, transverse, right? I think mechanical loading is axial direction, and then interestingly, in all the axial plate, you have a bone formations, and then in the transverse one also significant. Some in the transverse one is also significant, but in terms of plate rod, right, you don't see too much adding new bones in here, but you do have some in the transverse direction. Okay, in the transverse direction. So transverse rod, even overall rod is not increased, but the transverse rod do add in new bones in here. So therefore you added a lot of plates in here. Every directions, a lot of them is in the axial direction where you apply mechanical loading, right? And same thing for remodeling. You can see here, most remodeling is added in the plate. You can see here, significant, and not too much overall in the rod. So that's very much related to mechanical loading, right? Because mechanical loading can see the axial plates, axial rod, they actually subjected to significant loading in the compressive loading on them here. Okay, and then there are plates that have more axial one than the transverse and oblique. Okay, and in the rod, actually, it's interesting. There's not too much difference in terms of tensile stress and tensile strains, but in the rod, you can see they actually in the transverse one, they're receiving significant tensile strain as well. So, right. And so therefore, since the mechanical loading, compressive loading in the plates driven to the bone formation in the plate, in axial plate, and then the tensile strain in the transverse rods may activate in bone formation in some rod, but majority bone formation is at plate. So therefore, the plate rod is made because of they have different mechanical loading here. So in summarize, the increased anabolic modeling and remodeling targeting plate-like trabecula and also transverse rod-like trabecula. So the anabolic modeling actually is 3/4, and the remodeling 1/4 account of the mechanically induced trabecula. So now you can see modeling and remodeling not just doing the growth, there actually a process going on here. And the catabolic modeling are not affected by mechanical loading. So let me end up with a fun project. We're gonna study bone, right? I kind of mentioned, a little before in the vertebral structure, you've identified more than 85% of patients actually are Caucasians, right? And indeed, there actually are race differences between who's going to have fracture, right? So if you normalize Caucasian white by risk for fracture at 100%, you can see the Chinese about 30% relative to the risk in here. And then interestingly, Koreans and Japanese are falling between, even though we are very close to each other, right? So therefore, Asians have a low risk. And if you look at African Americans, they're probably all downweight in here, the much low have a risk of fracture here. So early on, we did a study many years ago now, is taking a premenopausal woman and looking at their bone structure. So in general, the Chinese are 2% shorter and 11% less weight. And we did the HR-PQCT on them. And then when the results come back, it's really quite interesting for us. So we did it in the tibia, in the radius, in terms of the plate and rod segmentation. As we presented here, green are plates, red are rods. You can see in the Chinese, you have a lot of greens than the one on the white, on the Caucasians, on the left side, right? If you quantify them, indeed, they have similar rod bone one fraction, but they plate bone one fraction in yellow in Chinese in here is double the amount in the Caucasian in white in here. And also almost double is the axial bone one fraction. And then same in the radius, same in the tibia, right? And then they are by numbers. Therefore, the Chinese women have significant plate-like trabecula in the structure. And those translating in significant dramatic difference between the mechanical property, right? So you can see the modulus is almost double from the Chinese, the mechanical property of the Chinese is double the amount than in the Caucasians. And so therefore they may explaining, why the Chinese women have low risk for osteoporosis fractures. And then now we including all ages, right? And then very, very interesting, independent of all the factors. And then it turns out we can just look at the bones. Can able to differentiate in Chinese from Caucasians, right? Plate rod ratio, differentiate Chinese. And then if plate rod ratio is less than 0.66, 86 of them correctly identify them as white. And if plate rod ratio greater than 1.3, and we identify correctly, 81% of them are Chinese. And then the ROC curve, is actually 0.780. So that's a very good predictor in terms of tell who is Chinese, who is Caucasian, right? So I want to do the studies ongoing is a study in China because we're getting me interested in regional ethnic differences in the different groups in China. And they will still want to do the study in China, in the US, but we have not able to do that. It's looking at American born versus China born, and also looking at how the loss in the bones in the during the period of menopause. And then it's getting my interest in genetics and anthropology here, right? I want to go back to this one, right? White have much higher risk than Chinese, but it's interesting Korean and Japanese distributing here, right? And then now we know where everybody come from. We all come from Africa. All the Asians actually come from Africa, right? And then there was several migrations. The yellow one is a little earlier. It's about 20,000 years ago. And those people, they last of ice ages to walk to Japan, yellow in here, they settled in there, they become Japanese. And the second wave, probably more than 5,000 years ago, that's really the people end up in here in blue or Korean. But you can see they are within 10%, 20,000 years, and then Koreans about, less than 10,000, 6,000 years of history. And then you can see, well, pretty close to each other. You can identify people in here and how the Koreans, Japanese, and Chinese bone risk will be different. I think I'm very fascinated in here. My hypothesis is the bone structure different. So we're not there yet. But if you go into red arrows in here, this is also from China. And then they are very close to Caucasians. The reason of them is there are 86% genes is Caucasian. They're Uyghurs. So the minority group in here, in this northwest territory of China, they have multi-race in this province called Xinjiang, but also have a lot of Uyghurs we call in here. So they are unique population. I want to do the study. I want to compare them to the regular Chinese folks in China. Difficult to get people to study here. So I'm pitching a dream to them. So if you build it, they will come. So I say, I'm going to build a multidisciplinary genetic medicine institute. And then we do independent disciplinary groups, recruiting patients, genetic typing, demographic, biochemical. I'm studying bone microarchitecture, doing osteoarthritis, and other people doing cardiovascular disease cancer can do. Not too many people do answer my call. So finally, I decided I need to go there. I really need to bring the measurements to the patients. So therefore, I able to convince somebody to give me money to build a super duper RVs in here, and then RV here, and then I have living quarters, and they have CTs in here, I have a bone density measurement here, and then the trailer can extend. So therefore, I have people sitting here, working here. Sorry, apologize, they're in Chinese. And that's the one you will build up in here. You can see here, that's the trailer. And then inside you have these HR-PQCTs in here, and they're pretending I'm driving, but I cannot drive those trucks yet. But hopefully next in 2 years, I could do my sabbaticals in there, I can do that. And then I send my students back after COVID. That's the trailer was sitting in the China now. And then that's where she's sitting. We have meetings talking about one here. So we're pretty exciting. And it got me very interested in the bone microstructure, right? I told you can differentiate into Chinese versus Caucasians. And they can also use in the strong structure to differentiate with a different species of the apes, right? So you can see if you do an analysis, you can separating humans and chimpanzee and gorilla and so on and so forth. And then the ITS actually can second it out. That's quite interesting by bone microstructure. So if you look at it here, that's anthropology doing, right? What they're looking at the shape of your teeth, and they're able to separate separating the Chinese, not Chinese, humans versus gorilla versus chimpanzee and pangolins in here. So got to be very interested. And come back to here is, I think you have microstructure differences in disease and different race. This probably also at the end also coming by the mechanobiology, because the mechanical biology actually driven what kind of plates and rod they're going to have, right? So therefore, going in a circle, we're interested in this revolution and the mechanical loading here. And indeed, it's quite interesting, as I say, fish have very dense bones. When they begin to frog and the mice, they begin to loosen bones. If you want to fly, you have less bones. And then the mammals decided going back to the ocean, then they actually have a reverse process. They now actually are able to gain bone back. So therefore, how the different genetics, different anthropology, and relate to bone microstructure is actually quite interesting for me. I'm going to end up my faculty in focus talking here. Thanks for my collaborators and my student. And thank you for spending Friday morning with me. I'm going to stop sharing. And if you have questions, happy to answer any question you may have. You may have, maybe there are no questions here. Lisa, back to you.
Yes, right now there are no questions, and but I have one question that it seemed, and maybe you'll say no, you heard that wrong, but it seemed to me like in disease state, or when there was development of bone disease, people would have more plates, like they lose rods and their plates would thicken. So that's sort of bad, and that would lead perhaps to osteoarthritis, because things get sort of thrown out of whack. But at the same time, the Chinese women had more plates. So is it sort of safe to say that if you sort of naturally have more plates, that's a good thing? But if you're aging and things are deteriorating and then you get more plates, then that's a bad thing? So it isn't necessarily a good thing to have more plates.
No, I think it depends where you're adding plates. You need a strategic adding in correct directions. So it is true. So if you look at the distribution of whole population, and in general, the people have very low density, they have risk of osteoporosis, they have good cartilage, don't have OA. And for the people actually have good bones, they actually have tendency to have OA. So that one is clear. But unfortunately, there are also significant people in the middle, and they have good bones, reasonable bones, and then they actually can develop in both OA and osteoporosis because of the location, right? They may develop OA in the knee, but they actually can have osteoporosis fracture in the spine. So I think there are also some people, my friends, that Tommy's raising his hand. So
yeah,
As our faculty, we probably Alexis allow Tommy to talk, or elevate him to as a panelist.
Or I can read his question. It's you can see it, Ed. It's in the chat.
Okay. Hey, Tommy, you can.
Yes, hi Ed. I enjoyed that very much. I was just thinking of other things, you know, with genetics. Of course, you're familiar with some of our relations, close relations to Neanderthal and Denisovan, and those components and different amount mixes within Eurasian populations that might have influence. But I was more curious about diet and latitude, the amount of sunshine, vitamin D that you would.
Tommy, I think the diet one, I think, you know, diet one, we cannot get rid of them because the Chinese, no matter where we go, we love Chinese food. Environmental will probably answer that question because this study, you know, we have done the studies compared the Chinese in Hong Kong and also the Chinese in Australia. The results are the same. So, for example, actually published paper already is compared Caucasians in Australia, the Chinese in Australia. The results almost superimpose each other. But the Chinese are, you know, at least the first generation, maybe the second generation, we love to eat the Chinese most time. So diet one, we need to tease out. That's why we actually want to do the studies in the US, is looking at, you know, the China-born Chinese and also American-born Chinese, and especially several generations later, right, to see whether there's any difference here. So, and then the genetics, I think is related to vitamin D mutations. Is we well know the African American, their body are not sensitive to any deficiency in vitamin D. They can have terrible vitamin D level, but they are fine. At least in bone, we know that. And about the Caucasians, the vitamin D deficiency causes a lot of trouble. I mean, in bone side is really, very bad for loss of bones. I think could be. That's just like we want to do this study, you know, do enough studies, analyze the genes to see whether that show up in these gene-wise studies. Is when we, you know, when the Caucasians evolve going to north, and then they struggling getting sunshine to get vitamin D's. So therefore, the light skin ones survive because they're stronger. They're muscle stronger, everything stronger, right? Then the mating, all the functions really depends on that advantage. I think that's what happens. Yeah, I think that's my wide hypothesis, may relate to vitamin D. But I need to do that study in China, so I'm still working on it to try to get some colleagues interested in China to be able to do this study.
So we're just about to wrap up. I have one last quick question about your van.
Yeah.
Will you also do blood testing in there?
Yeah.
Oh, okay. Yeah. So you'll get some of this information.
Yeah, exactly. We're getting either saliva or blood. You mean? Mostly right now we don't want the blood because we don't have people to.
You need a
nurse.
Right.
A bunch of graduate students, so therefore do a swab is okay. So we focus on swab first. So it was delayed, absolutely delayed by COVID. So we had a launch. We're supported by Columbia Global Fund to give us a planning grants, and then we launched these initiatives in December of 2019. So we immediately wanted the COVID, and then I was able to setting up my student in there. And then right now it's really figure out the logistics and figure out the IRBs, and because you know, and also figure out the you know, driving the van to different places, make sure everything's still working. So that's actually a big test we have in here because you're moving equipment to different locations, make sure that it will work. That's not guaranteed. Yeah.
Right. No, it's really exciting and ambitious. It's cool.
That's my plan when in couple of years, and then we're stepping down the chair, and then I intend to do my sabbatical in there, really setting up these studies. It's fun. Yeah.
Well, if you need a helper, I'll come.
Yes. I'd love to have my friends and colleagues travel with me, and because that's why I was getting a driver's license driving that truck in China. Then I really want just go to the steppes in Mongolia, go to these exotic places, and then park my minivan, no, not minivan, park my RVs in there. We can actually, you know, shoot in some, you know, deers or wolves, whatever. And then,
Wow.
yeah, in the meantime, we're actually also getting sun stuff. So it's my dream, and I intend to achieve that dream. That's why I call it a fun project.
Yeah, no, it's great. Good sabbatical
Yeah,
project.
because of it.
So
Thanks for so many people to join this link, and this is starting of a new series of BME Faculty in Focus. And then I hope my colleagues in our department can join this, and then we'll be on YouTube, and then we periodically update. Hopefully, in a couple of years, I'm going to update this video such that we have results of our exciting studies. Not, you know, in my, you know, other study research, as well as this we call the Silk Road, the Silk Road project. That's what I call it.
awesome. Well, thanks everybody for listening, and have a great weekend.
Yeah. Thanks, Lisa.